All right, so now we are going to pick up talking about vision, specifically about vision in our eyes. So, a couple of the key things we need to know about when we are talking about vision, hearing, a lot of our sensory information is that the sensory information that it comes that hits us isn't necessarily in a form that our body and our brain has the ability to detect. And that is the function of our eyes, the function of our ears, the function of our skin. We don't see with our eyes, we see with our brain. And it's this process of transduction, which kind of means to transform, which is the conversion of one form of energy into another. So for instance, with vision, it's converting the light waves from, that hit our eyes into signals that our brain can detect. So, a couple key terms that we need to know when we're talking about vision: wavelength, hue, and intensity. Wavelength is the distance from the peak to the peak of a wave of light. The hue then that we see is, the hue that we see then is determined by the wavelength of the light. So blue, green, red, those colors are all determined. How our brain registers them is based off of that wavelength. So hue is determined off of wavelength. And then the intensity of the color. So how bright the colors are is determined by the amount of energy or the intensity of the wave. So what then does that look like? First, when we talk about the visible spectrum for humans, obviously we can only, hopefully this is science stuff that you guys remember, we can only see a little bit of that, only from the 400 to 700 kind of range, where there's a lot of other light waves that are outside our brain's ability to see, to detect. So the physical property of waves, again, going back to this idea of wavelength, to determine hue, and then also the intensity. So a short wavelength, again, wavelength peak to peak. Short peak to peak distance is high, equals high frequency, which means our blues, the blue end of the spectrum. Long wavelengths, long distance from peak to peak, is our reds. And then amplitude, remember, is how bright the colors are. When the waves are very tall, we have the very bright colors, and when waves are very short, we have dull colors. So again, wavelength is peak to peak, and it determines the color that we see. And then the height or the amplitude of the wave is the intensity of the color, how bright we see. So our eye, we all love our eyes. We all hopefully don't take them for advantage of them, and they are, very fascinating in the sense of how much goes on with them. But again, a key kind of take-home idea: we don't see with our eyes, we see with our brain. Our eyes serve as this transduction. Our eyes are the structure that takes the light waves in and transforms that physical energy into a type of electricity that our brain can then register. So the parts of the eye that you need to know and understand: the cornea, the pupil, the lens, the iris, and the retina. Okay, so we'll walk through these piece by piece and kind of go over what everything does. So in the structure of the eye, the cornea. Okay, the cornea is the outer covering of the eye. It's the glossy kind of outside of the eye. If any of you have ever scratched your cornea, something like that, it can hurt. Okay, it can be very sensitive, but it kind of is a protective nature, protective structure over the eyeball. Okay, the pupil. The pupil is the adjustable opening in the center of the eye through which light enters. The little black part, it gets big when it's dark because it opens up to let more light in, and it gets small when it's bright, to let less light in. Okay, the iris, it's the ring around the pupil. The iris is actually a muscle. Okay, it's the colored part of the eye, but it's actually a muscle, and it's the part that lets the pupil open when it's dark and constricts when it's light. The lens. So right behind the pupil is this curved structure the lens. Okay, so the lens is kind of this transparent disc that sits right behind us, and it does something called accommodation. And accommodation is basically that it shifts. It goes like this depending on whether or not you're looking at something far away or close up. And the purpose of that accommodation is then to help focus whatever you are looking at onto the retina. Okay, the retina is located at the back of the eyeball. Okay, so the retina is, if you follow the cursor, the retina is this part all along the back of the eyeball, and it's the light-sensitive inner surface of the eye. It contains the rods and the cones, and plus all of the neurons that perform this transduction that we were talking about. Okay, in the middle is a bunch of fluid-filled, a fluid-filled kind of cavity. That's what keeps our eyeball, holds our shape. So again, focusing in on the retina. Okay, we have our rods and our cones. Okay, cones see color. That's our little mnemonic device there. See, cones, color. Okay, and rods predominantly see light and dark. So when we look at the rods versus cones in human vision, you can see that we have many, many more rods than we do cones. Okay. The location of the cones tends to be right in the center, and that's why we see things, we see color the best when we are looking directly at it. The rods are all in our peripheral, our outside and kind of our side vision, and that's why, you know, maybe you've ever noticed that you catch a flash of light out of the corner of your eye, but when you turn to look at it, it starts to disappear because, you know, you have more rods on this outside part of your vision that you can detect, you know, subtle movements and subtle flashes of light that, you know, your focal vision, your cones don't necessarily detect. Cones are not very good in the dark. So that's why we don't see colors as well when we are in the dark, but rods are better in the dark, or so they see better in the dark, and that's one of how our vision at night helps us. Okay, cones obviously are very color sensitive, rods very low. Cones are also detail sensitivity. That's why, you know, when you are looking, like, you know, when you're looking straight ahead, everything in your focal, your straight away is very, very detailed. And that's because your cones are seeing them. And then if you kind of just take a minute to pay attention, you can see that off to the side, things get a little bit fuzzy. And that's because the detail of the rods are much less. So just a fun little video clip here for you of the Blue Man Group, if any of you have ever seen them in concert. Pay attention to the words flashing on the screen.
Each of your eyes has over 3 million photoreceptors called rods and cones. These receptors convert light into electrochemical signals that travel through the optic nerve and into the brain. Here, these signals trigger the neurological process scientists call the hemorrhagism that happens inside your bristle. The rods in your eyes specialize in night vision and can function at much lower light levels than cones, but they do not respond to color. This is why we can only see in black and white when we are in the dark. The cones in your eyes, on the other hand, specialize in color and come in 3 types. Those that respond to the color red, those that respond to the color green, and those that respond to the color blue. All of the colors that we experience are the result of these 3 types of cones firing off in different combinations. something. The rods and cones in your eyes fire in rapid succession. But between each firing, there is a brief resetting period, during which your eyes are unable to take in any new information. Your brain covers up these microscopic moments of blindness with lingering after images, which help make your vision appear to be fluid and uninterrupted, even though it is not. This phenomenon, known as persistence of vision, is the unique physiological quirk that makes the illusion of animation possible. The dark spaces between each still frame of animation literally sneak by while your eyes are not looking.
Your eye, pretty cool. All right, so what happens when the light hits the retina? Well, the retina, the light comes in, right, passes through the lens. It's now focused, hopefully, on our retina. Now, what's kind of, well, not confusing, but something to really pay attention to is how this process works, is that the light comes in, okay, follow the cursor. The light comes in, it goes all the way through, okay, to the back of the retina. Okay, so let's see. It goes all the way. Let's see if I can get my pen to work here. Okay, it comes all the way through here, okay, to the back first, okay, and then it hits the back of the retina, and then it bounces back through, okay. So it comes back here. First we have our rods and cones, okay, so it comes and hits our rods and cones. Okay, then it goes back through the first layer of cells, okay, our bipolar cells, okay. Bipolar cells start this transduction process, okay, then they transition to the ganglion cells, okay, we have our, and then it travels along the outer edge of the retina into our optic nerve, okay, and then it travels out through to the brain's visual cortex and then to the thalamus. Okay, so again, important thing to recognize here, okay, the light comes in, passes through the retina, hits the back of the retina, bounces first to the rods and the cones, then the bipolar, then the ganglion, then out into the optic nerve, okay? So rods and cones, bipolar, ganglion. You need to remember that order, and you also really need to make sure that you understand that it starts at the back and then moves to the front of the retina, okay? So that's a really important organizational piece of the eye to know. So I get ahead of myself even when I'm doing it this way, guys. All right, so now the optic nerve is the point where the bipolar and the ganglion cells do this transduction process, okay, and then it carries the message in a way that's now readable into the optic nerve. The optic nerve then carries it through into the actual part of the brain. So where the optic nerve sits on our retina is what we refer to as our blind spot, and the reason being is there's no retina there because that's where the optic nerve is entering. So if we go kind of back and look at our other slide, you can see this little gap right here. This gap right here is our blind spot, because look, there's no retina, there's no bipolar, there's no ganglion cells, so it's not doing any kind of registering information. Now, the fovea is the central focus point in the retina, okay, around which the eye's cones cluster. So all of us in our eye have a fovea, and the fovea, okay, let's go back one more slide, okay, and the fovea is going to be the point on the retina where we kind of have the most cones, and this is where ideally your eye, your lens is going to focus the information, or focus the light waves so that you can see and see the information the best. So this little picture here, I think it's better if you do it in your book. It just kind of demonstrates this idea of the blind spot, and you have to focus here. But it's kind of hard to do on a screen, so I would suggest going and finding this in your book and trying it. Okay, so again, just to review, blind spot here, okay, where the optic nerve is on the retina. Okay, and then we have our fovea, the point of central focus. Okay, this is where the eye's cones cluster right here on the back of the retina. Okay, so this terminology is really important. You need to remember the details of each of these vocabulary words. Okay, and then the optic nerve carries it to the brain's visual cortex of the brain. So what does that look like? Well, again, so here we have our eyeballs. Now, again, something interesting about the brain and how the information is processed is that our brain is kind of crisscrossed. So our left eye actually is seen on the right side of our brain, and our right eye is actually, stuff that we see with our right eye is actually seen on the left side of the brain. Now, for most of us, we don't even register for this, we don't even pay attention to this, just because of the seamlessness of it, and plus the cross-visual fields that we have. And we have this neat little thing in the center of our brain called the corpus callosum, which allows the two halves to communicate. So the only time this actually, this ever becomes an issue is either, you cover one eye and you only see things on this eye, but again, because of the corpus callosum, right, our brain can see both, what our two sides of our brains talk to each other. It's when the corpus callosum is severed that we start to run into some really interesting things, and we'll talk about that in a unit or so. So again, if we look at the pathways from the eyes to the visual cortex, okay, it comes in here, it hits the thalamus, okay, because the thalamus, the sensory relay center, so all of our senses except for smell always go from the organ that detects them to the thalamus, okay, and then it gets to the visual cortexes in our occipital lobe, which is located at the back of the brain, okay, and that's where we actually see the information. So now, once we're in this visual cortex, what do we do with these light waves? How do we know what's going on? Well, our brain has things called feature detectors, and feature detectors are specialized nerve cells in the brain that respond to specific features of the stimulus, such as the shape, the angle, the movement, okay? So again, feature detectors are located in the brain. They're in the visual cortex of the brain. Feature detectors, visual cortex of the brain. That question shows up often on the test. So what these feature detectors do is they allow us to do some of this parallel processing. So we can detect color, motion, form, depth, all of these things at once. This parallel processing goes on simultaneously. So as we're coming in and we're looking at the features of a bird, for instance, we can detect the angle of the wings, we can detect the angle of the feet, we can detect the height, all of those kinds of things, as well as the color, the motion, the form, the depth. We can do all of this stuff at once. Okay, and this is kind of how our visual process works. So in terms of how kind of the cycle of it works, we look at something, our retina processes it, the rods and cones, the bipolar cells, the ganglion cells. Okay, then our feature detectors respond to angles, lines, edges, those types of things. Again, feature detectors in the visual cortex of the brain. Then the parallel processing happens, allowing us to determine color, movement, form, depth, all of those types of things. Okay, and then finally, based off of all of that information, our brain interprets and recognizes what we're looking at based off of information from stored images. Now, how does color vision work? Well, color vision is something kind of tricky, and we have two different theories on how we see color. The first one is the Young-Helmholtz trichromatic, or the three-color theory. And basically what this one says is that that the retina contains three different color receptors, okay? One most sensitive to red, one to green, one to blue. And then when stimulated by the different light waves, in any combination, we can produce the perception of any color. So the Young-Helmholtz theory focuses on the retina, the rods and the cones part of our brain, and says that the cones, we have these 3 color receptors that the light waves trigger in different combinations to allow us to perceive color. Okay, so this is oftentimes what we do when we're testing for color blindness. So for instance, when you look at this picture right here, most of us would say, okay, there's a bunch of, orange and green dots. The green dots say 74. Now, if for whatever reason you couldn't detect this 74, then we would investigate whether or not you had an issue with color blindness. Now, the opponent process theory is kind of the secondary part of color vision, and it focuses less on the rods and cones and more in the bipolar ganglion optic nerve visual cortex part of the brain. So this theory says that opposing retinal processes, it says that we have red, green, yellow, blue, and white, black, and that the activation of those enables our color vision. So if we were to, for example, some cells are stimulated by green and inhibited by red. So and others are stimulated by red and inhibited by green. So if we have a green light wave coming in, we're going to stimulate those green ones and we're going to see green, okay? Or if a red light wave comes in and it inhibits the red, then we're going to see green. Okay, so it's kind of a tricky color theory. Kind of a tricky theory, but basically it just says that it's the opposing retinal thing. So we have red, green, yellow, blue, white, black, and that the opposite process is what we see. So this is kind of where we get the after image effect from. So take a look at this picture. Okay, what I want you guys to do is stare at this. We're going to stare at this for about 30 seconds. Okay, right in the center, try to stare. You can blink, that's fine. Just try to stare right here in the center. And then I'm going to put up a white screen, and I want you to tell me what you see. All right, so we're going to say 30 seconds, go. And what do you see? Okay, now hopefully most of you should be seeing the after image of a red, white, and blue flag. And again, when we're going back to this idea of the opponent process theory, so because we stared at green, yellow, and white, and those, so according to the color process theory, green, yellow, and white kind of burnt out, right? So when those were burnt out and we went to the blank screen, green, yellow, and white were no longer working, so we saw red, white, and blue. Oh wait, no, I said that wrong. Yeah, green, black, and yellow, green, black, and yellow, and then we saw red, white, and blue. Sorry. All right, now what about motion? Okay, how does motion work? So let's take a look at this and kind of just, how long does it take you before you see movement in this illusion? Probably not very long. Probably, you detect some motion along the black circle here. You probably see some motion here. You probably see some motion in here. Okay, so now why do we see this motion? Well, we see this again going back to this idea of the color burnout. Our eyes are all over the place and we're constantly getting the activation and deactivation of those cells based off of the light, the white, and the black. And so the burnout and kind of that after image that we see is giving us this illusion of movement. Okay, now if you get migraines, if you have seizures, anything like that, don't do this. This isn't a big deal. Okay, so what's going to happen is we're going to stare at this center thing. The circle is going to start spinning. Okay, you're going to stare at this center dot, okay, for approximately a minute, and then a photo is going to come up on the screen. So we're going to see what happens when the photo comes up. So what do you think is going to happen? Okay, I want you to choose A, B, or C in your notes. Okay, just kind of jot down real quick. Do you think the photo is going to appear to rotate counterclockwise? Do you think the photo is going to appear to rotate clockwise? Do you think the photo will appear to expand? Or do you think the photo will appear to shrink? All right, you got your choice? All right, so we're going to stare at this for approximately a minute. Ready, set, go. And stare at the photo. What do you see happening? So you should see the photo kind of seeming like it's coming at you or kind of pulsating, okay? Some people, don't see anything, it doesn't work as well, and other people see a very drastic event. But again, so you should see the photo look like it's expanding, coming towards you. And why does that happen? Well, when we go back and we look at our thing, when you're staring at it, it feels like that center dot is moving away from you. So when we stop it and we look at the guy, our eye now is kind of reevaluating and it has to readjust to the fact that, we're not getting an illusion of movement away, so it looks like it's moving forward. Our eye is kind of like pulling itself back to normal, our lenses. All right, so that's it for today. We'll see you guys tomorrow.